Hello to our friends joining us via recording. We are discussing lesson number 2 for lecture. This lesson is about cells. So one of the first things we're talking about with cells are the organelles that are found inside cells. Here's a reminder from something that we said in yesterday's class, that you are responsible for knowing the function of the organelles that are in the cell. And the best place to reference that is going to be your lab packets. So there's some information there for you in the guided lesson. There's some videos for you. You've also got those lab packets that'll help you out with what the organelles do. To help us with figuring out some of the things that a cell needs to be able to do, we decided to make a list of some of the things that we've needed as we have been surviving quarantine. We were joking in the chat that it's been 10 months that we've been stuck at home, only 10 months, if we can believe that. But we made a list together. We kind of compiled our ideas about some of the things that we absolutely have needed to get through these last 10 months together. So looks like some of the biggest things on my list, no surprise here, things like food, right? So whether it's grocery pickup or still going to the grocery stores, we've definitely needed some food sources. When we think about a cell and we think about the food for a cell, food gives us energy. What organelle in a cell makes energy? Who's doing the, shall we put it in these terms, who's doing the cooking? Who's making our energy for that cell? Yeah, I'm kind of slow on my internet's a little laggy over here. That's absolutely right. Lots of us are chiming in that the mitochondria is what makes the ATP, or it's what's cooking up that energy for us. So just like we've needed to have food, some of us have needed to learn how to cook, right? Since we're at home so much more. Same thing with a cell. The cell uses its mitochondria to take ingredients like its favorite food, that's glucose, and turn that into ATP, the energy that it has. Looks like we've got a couple of things on here that were big as well. Things that would help us with exercise, for example. So things like biking. I'm super excited because we have been waiting since Thanksgiving on an elliptical. We finally have an elliptical that is almost ready for us to use. So I'm not on the biking train, but I'm going to try to do the ellipting train. So things like biking, things like being outdoors to get a little bit of stress relief, right? Or being with our family. Although I think some of us could argue, right, that we're having to survive our family in quarantine, right? If we're being honest. Speaking for a friend, not for myself, obviously, right? We have on our list here the membrane, and that is a cell thing, right? A cell structure. But I think that this membrane ties in really well to when we're thinking about our shelters, or the place that we've lived, the places that we've stayed during this quarantine time. So a cell is sheltered or divided from its environment using the plasma membrane. So the plasma membrane decides what can come in and come out. Not everything can get in and out of the cell. That's because the plasma membrane says, I get to choose who comes in and out. I'm really glad, by the way, that at least one of us put into our thing here, masks. Masks have been essential, right, to making sure we don't catch COVID. Masks are also acting a lot like the plasma membrane of the cell. So deciding what can and can't get into your airway. If we put a mask on it, stuff can't get in as easily. If we take the mask off, stuff can. So having masks have been essential for going to those grocery stores. If we're not doing grocery pickup, if we're not doing Amazon and having groceries delivered to us, being able to go out safely with those masks. And I also see in here, and I love this, I saw chopped up essential workers. Hey, here's a shout out to the essential workers that are here in class with us today. If you are an essential worker, can you give me a wave in the chat if you're considered an essential worker? That can be medicine, that can be grocery store, that can be anything where your job didn't get to stop. You don't get to work at home. Yeah, look at that. We got some people here. Here's my shout out to all my essential worker friends. Truly, thank you for what you're doing to keep us going, to keep us safe, to put yourself and your family in the line of duty. We really appreciate you. So thank you to all of my students who are essential workers, to all of you who have family that are essential workers. We appreciate you. So I'm not quite sure how we can relate that to an organelle in the cell, except to say that a cell needs its neighbors to be working well for it to work too. So to all my essential worker neighboring cells, I appreciate you and am grateful for you. One of the things that we didn't specifically put on our map here, but it's something that I think we all kind of maybe take for granted, is the fact that we need air to survive. Inside our houses, hopefully, or our apartments, hopefully it's been clean air. We're not too much virus floating around. Our homes are filled with air. Does anyone remember from working on this learning activity what a cell is filled with? We actually chatted about this yesterday too. What was the name of that stuff in a cell? Yeah, so what fills up a cell, kind of like the air of where we live, the air inside a cell is called cytosol. So it's the soupy stuff that's inside the cell. The cell uses it to keep its shape. It uses it to store some of its food. We use air to help us breathe. So the cytosol is kind of like the air that's in our environment. We've also definitely had a lot of hand sanitizer, right? Or it's kind of funny that what didn't make our list here is toilet paper, right? The great toilet paper shortage of March and April. Those help us to survive too. Things that keep us clean and keep us healthy. That's kind of like maybe those peroxisomes that detoxify and get rid of stuff, like hand sanitizer. Lysosomes that are kind of like recycling we talked about. So we've continued to have trash service, kind of like our cells have lysosomes. So as you, my challenge for you would be as you're studying the organelles, to make it a little more fun than just memorizing them, consider how quarantine, or consider how being stuck at home has made you rely on certain things, and see if you can relate that to any of the organelles in the cell. So thanks for giving me some ideas of what got you through. 100%, I'm totally there with you on the internet, on delivery stuff, on Netflix. Although I'm going to be honest, with my two littles running around, there has not been nearly enough Netflix. It's really sad. So enjoy, yeah, and Barbara mentions her Bible, lots of different things too. Our spouses, our boyfriends and girlfriends, those things that fill you up, not just food-wise, right, but the things that help you feel a little bit better. Definitely keep relying on those as we continue with our quarantine. Hopefully not forever, right? Fingers crossed. So we will see. All right, let me do one more poll just to get an idea really fast. Can you all help me out with how much of this lesson that we've done? So you should already be in my poll. If you sent me anything else, if you texted me anything else, all I need from you is a letter. A, B, C, or D. How much of this lesson have we had a chance to look at? Because that's going to help me know where we stand. Yeah, that's okay, Barbara. It's perfect if you put those in there. So all right, I got about half of us. Remember, it's completely anonymous too. So if you're down there at D, you haven't done anything yet, that's totally fine. I will not know it's you. Oh, weird that it's saying that because that the other poll was maxed out, but this one shouldn't have been. Well, I'll see what we got in and we'll call it good. Okay, awesome. So most of us have at least started on us. And for any of my friends who are down here, that's totally fine. We will not be able to cover everything perhaps in the depth that you would want to in our class time together if you haven't started yet. So recognize that if you haven't had a chance to start or haven't done very much yet, there's still going to be a lot of work for you to do after today's class, but I'm going to try to hit some of the trickiest concepts to help make sure that we really understand what's most important from this lesson. So props to all my friends who have completed all of it or about half of it or even just started on it. That's perfect. We'll get all of us up to about halfway there. That's the goal for today. All right, so the first thing that I'm going to start us with in our class today is we are going to look at the names of the types of fluid in the body. So when we're talking about fluid, you are so much fluid. A lot of your body is made out of fluid. But that fluid doesn't all have the same name. We name different fluids in the body based on their location. So as you're studying the fluids that we find in the body, make sure that you're not only learning their name, make sure you're also learning how to describe where I would find them. So at the beginning, when you're thinking about fluids, there are two places that you find fluid in the body. So big picture, either we have what's called intracellular fluid, or we have, I'll add another one under it, what we call extracellular fluid. Every fluid that we're talking about today, every fluid that we'll talk about all semester is either going to be an intracellular fluid or an extracellular fluid. Now, to help you remember what intracellular and extracellular means, I want you to think about an extraterrestrial. And there's one in particular who's pretty famous. Does anyone know the most famous extraterrestrial who I might be thinking of here? Yeah, exactly. E.T., extraterrestrial, E.T. If we were in lecture class together, I'd totally pop up a picture of him. I don't have one right now. But E.T., he is an alien, an extraterrestrial. What this term extraterrestrial, oh, I love it. Jessica's husband knew it was E.T. Props to you, Jessica's husband. That's great. E.T., he's an extraterrestrial. And what his name literally means, extra means outside, terrestrial means Earth. So an extraterrestrial is some kind of being that is outside of the Earth, whether it's an alien, or I always like to point out too that not all extraterrestrials are aliens. When we send astronauts to the space station, they are extraterrestrials too. They're outside of the Earth. So extraterrestrial or extracellular fluid, extra always means on the outside. So when I talk about extracellular fluids in the body, I'm talking about the kinds of fluids that I find outside of a cell. There are a lot of different places in your body that you have fluids that are outside of your cell. That's why they're going to have lots of different names. We'll talk about them. Intracellular fluid is the other kind of fluid that I have in the body. Let's take a guess. I promise it's not a trick question. This intra part right here, what does intra mean? What do we think? Intracellular. Yeah, exactly. Intracellular, intra means inside of. So all of us, as we sit at our computers right now, we are all intraterrestrials. We are inside of Earth. Intracellular fluid is the kind of fluid that is inside cells. So first important point for us to keep in mind throughout our whole lesson number 2 here. Every fluid in the body is either an intracellular fluid, it's inside a cell, or it's an extracellular fluid, it's outside a cell. These are my 2 big picture categories. Make sure that you can sort everything into one of those categories. Now, the good news is there's only one type of intracellular fluid, and we've already said its name today. What's the name of the one kind of intracellular fluid? There's just one. Yeah, the one kind of intracellular fluid is called cytosol. Anytime you hear cytosol, you should be thinking this is the fluid inside a cell. And it doesn't matter if we're looking at a cell like would be in tissues. Doesn't matter if we're looking at a red blood cell. All of your cells have fluid inside of them, and the name of that fluid is cytosol. So my one intracellular fluid is cytosol. It's found inside every one of my cells. When we start talking about these extracellular fluids, now we're talking about all kinds of different fluids that are found in all kinds of different places in the body. So let's describe some of these extracellular fluids. One of these extracellular fluids is called plasma. When we talk about plasma, plasma is a type of fluid that we find outside of red blood cells because it's extracellular. It's outside of our red blood cells, but it's inside a blood vessel. Inside a blood vessel. If you ever donate blood, the red blood cells, which is what we're trying to give to someone else, or those actual cells, they're going to be suspended in plasma. That's the fluid that's around them. Hey, there's actually a lot of talk about plasma right now related to COVID, right? For people who have had COVID and who have recovered, they can actually donate their plasma because this fluid that's outside of the red blood cells, it has a bunch of antibodies, or it has a bunch of proteins in it that would fight against the coronavirus. So plasma in the news these days, when we're talking about how to fight off COVID-19, we make proteins inside plasma that are part of our immune system, our immune responses. So outside of your red blood cells, inside your blood vessels, we've got this plasma fluid that has those antibodies, and that's going to have a whole bunch of ions that we're going to talk about today. Outside of cells, inside a blood vessel, I call you plasma. Hey, if you live outside of cells, but you are inside a tissue, I call you interstitial fluid. Interstitial fluid. This is the kind of fluid that's outside of my cells, so I can see it here in blue, but it's inside a group of cells together. We call that interstitial fluid. When we're thinking about the types of fluid in the body, there's a lot more interstitial fluid than there is plasma. But both of these types of fluids are found outside of our cells. Both of them are extracellular. So, big pictures we've talked about so far. Make sure we know the difference between intracellular, inside a cell, and extracellular, outside a cell. Make sure we know where to find the cytosol, the plasma, and the interstitial fluid. Great question in the chat, asking about purple dots here that we're looking at. Yes, these absolutely are the nuclei of these cells. Let's see if we remember from yesterday's lab class, what's that dark spot in the middle of those nuclei? Anybody remember what that was called? Yeah, so the little dark spots that I see in the middle of the nuclei are called the nucleolus. Absolutely. It's kind of like the little nucleus inside the nucleus. So in this picture, we are seeing a cell filled with cytosol, and we're seeing that big organelle in the middle called the nucleus with its little nucleolus inside. Great connection to make. Absolutely. When we talk about the fluid in the body, not all fluid levels are equal. And this is a pie chart that shows us about how much of each kind of fluid we have in the body. Now, every semester, my students very sadly remind me that this is not actual pie. So that is very sad. Know that I'm grieving that with you. But my pie chart here, if we pretend that it's pie, the size of the piece of pie gives you an idea of how much of something that we have. So if you're getting the really huge piece of the pie over here, that's intracellular fluid, meaning the fluid in the body that I have the most of is intracellular. What was the specific name of intracellular fluid again? We didn't just call it intracellular. We had a technical name for it. Yeah, absolutely. Intracellular fluid, the one example, we called that cytosol. So cytosol is the fluid in the body that you have the most of, meaning that over half of the water that's inside your body is found inside your cells as what we call cytosol. Notice my next biggest piece of the pie, shown here in blue, is the interstitial fluid. The interstitial fluid is my first example of an extracellular fluid, interstitial fluid. So interstitial fluid is outside of my cells, but it's inside what? Where is interstitial fluid found? Yeah, this is the one that's inside tissues. Oops, I spelled that wrong. Inside tissues. And we're going to start talking about tissues next week in lab. But if you want to think of a good example of where you'd find interstitial fluid, when you look at your forearm and you see the skin and you feel the muscle underneath it, the fluid that's in between those things, that's a good example of where you'd find interstitial fluid. Interstitial fluid is basically everywhere in the body, but if you want a place you can look at it, just look at your forearm. It's there inside your forearm. It's there inside your legs. It's in between your cells everywhere. Plasma was not found everywhere. Plasma was found in one place in the body. Do we remember where that one place was again? Where's my plasma found? Yeah, plasma is that fluid that's inside your blood vessels. So it's outside of the red blood cells. It's the liquid part of your blood. Notice that I have this little tiny sliver over here that's labeled other fluids in the body, other fluids. Ironically, this little sliver of fluid here is where I have a ton of different kinds of fluids that I find in the body. So your guided lesson talks about several of these different fluids, but let's mention some of these fluids. One of these fluids is called synovial fluid. Where would I find synovial fluid? That's one of my random others. Synovial fluid. Yeah, exactly. Remember how we talked about synovial joints? How they had fluid in between the bones to help the bones to move. So that's one of my other fluids. I don't have a lot of it, but I need it. It's still important. We've got some stuff called lymph fluid. And lymph fluid is going to be found in a similar place to plasma. You know how we said plasma was found in blood vessels? Well, lymph is found in the lymphatic system's version of a blood vessel. So we call it a lymphatic vessel. These are the connections that go between the lymph nodes in your body. So a lot of your immune system cells, they use these lymphatic vessels as a superhighway to travel throughout your body. The fluid in between them is lymph fluid that we find inside these lymphatic vessels. I see your question, Fanchin. Give me one more moment and I will get to it. One last kind of fluid that I want to mention really fast, cerebrospinal fluid. Cerebrospinal fluid. What is this fluid found around? What organs do I find this stuff around? I'll mention too that we know it as CSF, cerebrospinal fluid, coming back later. Yeah, exactly. So cerebrospinal fluid, the cerebro part of its name means the brain, and the spinal part of its name means the spinal cord. So we will talk more about cerebrospinal fluid when we get to unit number 3 and unit number 4. We'll talk about it twice. When we think about lymphatic vessels, we aren't going to talk much about these. You'll talk more about that in AP2. But we've already talked about synovial joints and synovial fluids. So this category here, we've listed some of the fluids that would fall into this category. There are others. I'm not going to require you to know all the different fluids in the body, but here's the big idea with extracellular fluids. Depending on exactly where you find it, it's going to have a different name. So where I find interstitial fluid is a different place than where I find cerebrospinal fluid, which is a different place from where I find plasma. But all of these extracellular fluids, all of them are outside of the cell. So outside of the cell, all kinds of different names. Inside the cell, just one name, and that one name is cytosol. How are we feeling about the locations of our different fluids? Thumbs up, thumbs down. Perfect. Lots of thumbs up. Okay. Well, let's take these locations then, and let's talk the next thing we need to know with these fluids. The next thing that we need to know with these fluids is we need to know what I call their recipes. So what I mean by this being the recipe is if you wanted to make each of these kinds of fluids in the body, what would you need to add to it? So yeah, we're doing cooking stuff again, right? We didn't get our pie, and now we got a recipe for something that's really probably not going to taste that great, where I'm just ruining all the cooking stuff, right? So let's do a recipe together for, let's start with intracellular fluid, because that'll be a little bit easier for us. When we talk about intracellular fluid, we're going to look at our bar graph here, is going to be shown in this beige color. So when we talk about intracellular fluid, we're looking for the bars on this bar graph that are much higher in beige than they are in blue or red. When you're looking at our graph down here, which of these things listed here are really high in beige compared to blue or compared to red? What do we see on this chart that looks much higher inside the cell? Yeah, I'm making you type a lot, aren't I? I'm being the mean teacher. Y'all are doing great. That's perfect. The first thing we noticed is check out this tall bar right here. So this tall bar right here represents K+, which, as many of us put in the chat for me, K+ is potassium. Potassium. By the way, I'm not going to make you memorize a lot of elements, but this is one that you do want to make sure that you know. So we want to make sure we know that K is potassium. This is going to be really important when we get to lesson number 4. We're going to talk all about K. We're going to talk all about Na. So make sure we know that K is potassium. When we look at our recipe for the different fluids, the one fluid that has a ton of potassium is intracellular fluid. Lots of potassium in there. The other thing that several of us mentioned in the chat that's also totally correct is that we have a lot of proteins in intracellular fluid. There's a lot of proteins here. And when you're working on the guided lesson, it talks a little bit about those proteins. So those proteins are going to do things like help a cell have its shape. They're going to do things like make chemical reactions happen. So lots of proteins inside our cells. We can see that bar is tallest when we're talking about intracellular fluid. So let's circle that here. Proteins are the other thing that I've got a ton of inside my cell. Now, I believe in your notes packet, you actually have 2 statements. Mine is here just in one, but I think you have it split up, interstitial fluid and plasma. Is that right, that we have those split up? Okay, perfect. Yeah, so I'm using an old version of my slide here, so we'll do a little funny business here to make it work. When I am looking at my 2 extracellular fluids, so interstitial fluid and plasma, there are a couple of things that both interstitial fluid and plasma are really high in compared to my cytosol. What are the 2 things that both interstitial fluid and plasma have a lot of that cytosol does not? Yeah, perfect. Several of us are typing it out for me. You can look at the size of the bars. Notice how I've got a blue and a red bar up here, sodium and for sodium, and I've got a blue and a red bar up here for chloride. Both of my types of extracellular fluid have a lot of sodium, that's Na, and chloride, which is Cl. So let me write their names underneath. Sodium is Na, chloride is Cl. It doesn't matter if you're interstitial fluid or if you're plasma, you're always going to have a lot of sodium and you're always going to have a lot of chloride. So keep that in mind for all of them, all of my extracellulars, sodium and chloride, lots of that on the outside. When we're talking about plasma in particular, notice that plasma also has a lot of protein. So on your sheet where you have it broken up, you would also say that plasma has a lot of proteins. Interstitial fluid doesn't have as many, but when we're talking about plasma, we do have a lot of proteins in plasma. And some of those proteins were what we were just talking about related to COVID. So things called antibodies. Let me type those out here. Antibodies. The job of antibodies is for your cells to grab onto something that's not supposed to be there so that they can shut it down. They can eat it or they can destroy it. Antibodies go and attach to things to let your immune system know what's not supposed to be there. So antibodies are a lot of those proteins. There was one other thing that we mentioned that these proteins in plasma do. Does anyone remember what else we said those plasma proteins helped your body to do? Something that we're grateful for when we get a paper cut. What do we want our blood to do? Yeah, exactly. We want our blood to clot. So another kind of protein that I see inside plasma are things called clotting factors. Clotting factors. Okay, so let's summarize what we just found on this graph. The first thing that we found is if I was trying to make intracellular fluid, if I was trying to make the liquid that's inside my cells, cytosol, what I would need a bunch of is potassium and proteins. Intracellular fluid, whole lot of potassium, whole lot of proteins. Maybe I don't want to mix up the stuff that's inside the cell. Maybe I want to mix up the stuff that's outside. In particular, I want to mix up the stuff that's outside in tissues. If I want to make that stuff, I'm going to make sure I've got a bunch of sodium and a bunch of chloride that's in that fluid that's outside the cells. Or if I want to make the extracellular fluid in blood vessels, I would have a bunch of sodium, a bunch of chloride, and those proteins that we talked about. As you are studying this material for your assignments and for the exam, make sure that you can write the recipe or what you would mix together to make each of these fluids. A really great way to remember the recipes for these fluids is what's called the salty banana. The salty banana. I wish that I could take credit for this, but this idea is not mine. It actually came from a guy named Mr. Anderson. I think we're going to watch some Mr. Anderson videos in lesson number 4. So he's the one that came up with the salty banana as a way for you to remember where we have a lot of different ions. So when we're thinking about a salty banana, your cell as a salty banana, let's start with what ion bananas have a bunch of inside of them. Why do we eat bananas? What do bananas have a lot of? Yeah, bananas have a lot of potassium. Potassium, the chemistry name for it is K+, right? So when I'm thinking about a banana, you eat a banana for the sake of increasing your potassium. Lots of potassium inside of there. The other reason you might eat a banana, though, is if you are diabetic. Why might someone who's diabetic eat a banana? What might a banana have in it that would help them? Yeah, exactly. Like Kaylin's mentioning. The other thing that bananas have a lot of is glucose or sugar. So if someone's having like a diabetic crash, they can eat a banana to help raise their blood sugar really quickly. It didn't make my recipe page because it wasn't on the chart last time. But the other thing that's really high inside cells is sugar, glucose. I can't remember if we've talked about this yet or not. Why do cells want to have a lot of glucose? What's glucose good for? Yeah, Audrey mentioned it. Glucose is my energy source. This is what a cell breaks down to build ATP. So when we're talking about what's inside a cell, cells have a lot of potassium, or inside a banana, excuse me. Bananas have a lot of potassium, just like our cells. Bananas have a lot of glucose, just like our cells. And we're going to pretend that our salty banana cell is kind of like the perfect banana, because the perfect banana would also have proteins inside of it. See, human cells have a whole lot of proteins inside of them. If we're a salty banana cell, we're going to pretend that we're a super banana, and we've got lots of proteins too. Okay, we've talked all about what's inside this salty banana. Now we're talking about what's outside our salty banana. And what's outside the salty banana is the two ions that make up salt. What's the chemical formula for salt? Does anyone know from chemistry? What makes up salt? Yeah, exactly. Salt is, if we want to put it in the chemistry terms, it's NaCl. It is sodium chloride. Sodium chloride. When we're talking about a salty banana, I took a banana, I sprinkled salt on it, and I have generated a cell. So think about, as you're memorizing your recipes, that we have a salty banana cell, meaning we've got something that has a bunch of potassium, a bunch of glucose, and a bunch of proteins inside of it, and we sprinkled some sodium and chloride on top, and we've generated a salty banana. So we must learn the salty banana. We should learn it now, because if we don't learn it now, it's going to come back to haunt us like nobody's business in unit number 3. So the salty banana, it's a way to remember that sodium and chloride, there's a whole lot of those outside, and there's a whole lot of potassium and glucose and proteins on the inside. Hey, so who wants to eat a salty banana? Any takers? I did this with my students at the end of last semester. It actually wasn't that bad, to be fair. Although I did have a student who brought an old banana, and she was like, this is terrible. So maybe at the end of the semester, we can do the salty banana challenge again together. We can try sprinkling a little salt on it. Yeah, Gerardo's like, hey, if it's a super banana, sure. Yeah, let's see if we can find a super banana, right? We'll look for some bananas that have some proteins. Make it happen. Nice. Christian's got bananas right now. Look at that. That's great. I don't have any good bananas in my house right now. If I were to do the salty banana challenge with one of the bananas I had, it would not be fun. So I'm going to take a pass for today. But maybe at the end of the semester, we can do it together. That'd be fun. All right, so you have this picture in your notes. This picture takes the recipe that we just talked about with that bar graph, and it takes what we just talked about with the salty banana, and it puts it into a picture of a very simple cell. So one of the most important parts of a cell is its plasma membrane, because that's going to decide what can get in and out of the cell. The plasma membrane, here's kind of a teaser for us. The plasma membrane is what makes it possible for there to be different concentrations of stuff on the outside compared to the inside. The fact that I've got a bunch of potassium on the inside and a bunch of sodium and chloride on the outside, that is due in part to the fact that these things can't get across the plasma membrane. We will talk more about this in the next lesson, but the fact that these things have a charge, so this one has a positive on it, this one has a negative, here's another positive. Anything with a charge is what I call an ion, and ions are not able to get across the plasma membrane. So potassium stays inside, sodium and chloride stay outside, unless I make them a special tunnel to get through. And that's where we're going here in unit number 1, talking about their special tunnel. In normal circumstances though, pretending we don't have any tunnels, we see a lot of potassium on the inside, a lot of sodium and chloride on the outside, and the number of these things is different inside compared to outside. So we actually end up with a different charge when we compare the inside to the outside. I won't make you learn a lot of numbers, but here's a number that we do need to know. And this number might be in a later lesson for you, but we can learn it right now. Negative 70 millivolts. Negative 70 millivolts. This is a number that we absolutely must know. This number represents the difference in the charge inside of a cell compared to outside of a cell. The inside of a cell looks like -70, or it looks more negative than the outside of the cell. There's more positives outside than there are inside. That leads to a resting membrane potential of -70. So that's probably new information for you. Don't freak out if you hadn't seen that number yet. We will talk about it more, I promise. But when a cell is at -70, when there's a different charge inside compared to outside, we would say that that cell is polarized. Polarized. And hey, this is an underline, highlight, star word, meaning this is a word you need to make sure you know what it means. Underline it, highlight it, star it, whatever you do to know what to study. It's a good thing for your cells to be polarized. It's good for there to be a different charge inside compared to the charge outside. If a cell completely loses the difference in charge, if it's the same on the inside and the outside, most likely that cell is dead. It's really important for your cells to be polarized. And the fact that we have sodium and chloride on the outside and potassium on the inside is one of the reasons that we are polarized. So make sure one of the things that we take away from today's lesson is that my cells are polarized. They are not the same on the inside and the outside. They are different, and different is good. So that recipe for the fluids that we made, that's why it's important for us to know how I make these fluids. Because the fluid that you put inside your cell has a different recipe than the fluid on the outside. And by having that different recipe, that helps the cell to stay alive and to do its job. So again, underline, highlight, star, polarized. When a cell is different on the inside and the outside. And different is good. Different helps my cell to be alive. When we think about cells, we said that cells are different on the inside and the outside. And that brings us to a discussion of what I like to call the tonic words. The tonic words. So you'll hear me say that a lot today. I'll probably say it again throughout this unit when we're working on stuff. When we talk about two different solutions, whether it's a solution that's inside the cell and in the tissues, for example, or inside the cell or in the blood vessel, when we're comparing two different liquids to each other, we can use tonic words to make those comparisons. So let me explain what I mean by these tonic words here. This ending part, tonic, just refers to concentration. Concentration. When I'm comparing two fluids to each other, there's kind of three options for looking at their concentrations. Option number one is my fluids could be isotonic. Isotonic. For my friends who had a chance to work on this lesson, what does it mean about the concentration of my fluids if they are isotonic to each other? What does isotonic mean? Yeah, exactly. A couple of us are chiming in here. Isotonic means that the concentrations are the same. Same. So if I had the same amount of potassium on the inside compared to the outside, I would say that my fluids are isotonic. The concentrations are the same. But in the body, our cells are polarized, meaning I have different concentrations inside versus outside. So let's talk about the concentration words that we use when we are not the same, when the concentrations are not the same. So let's talk about our first one here. Our first one is what we would call hypertonic. When I use the word hypertonic, what does that mean? If one solution is hypertonic compared to the other. Yeah, hypertonic means that it's higher or it's more concentrated. More concentrated. I like to remember hypertonic by thinking about when my daughter gets more sugar, she is hyper. She goes nuts. So hypertonic, there's more of something. There's extra. It's above normal. We've got an extra concentration. So too much of something. Compare that to the opposite, which is hypotonic. Hypotonic. If I talk about a solution being hypotonic, hypo literally means under concentrated. Or if we want to put it in like normal person words, we would say low concentration. Low concentration. If I look at a solution and it has less than the other one, I would say it's hypotonic. If I look at a solution and it has more, I would say it's hypertonic. If my two solutions are the same, we would say that they are isotonic, the same. So we live in the South. I thought a good example of talking about these different kinds of solutions are looking at the two types of tea. So here, let me do an informal poll. Let me build it really fast. I want to know, based on those of us who are here, what kind of tea we like. So you should see popping up on your screen at the very bottom, down by the person who's raising their hand. There's a little chart, and that is my poll. I want to know, if you were drinking tea, which kind would you be drinking? And if you're not a tea drinker, I'm not really a tea drinker either. Tell me which one, if I made you drink some, which one do you think you'd drink? I should have put an other option, right? The South has really gotten to me. I'm starting to just assume that tea goes everywhere. All right, I'm not surprised. We haven't quite all voted, but a lot of us have. Overwhelmingly, we do love our sweet tea. So sweet tea seems to be the favorite. Yeah, Audrey likes the Lipton kind. That's awesome. Gonna be honest, I mean, if I had to drink it, I would probably do sweet tea, but I'm way more of a coffee person. Or let's be real, I'm way more of a Pepsi person. Just gonna be honest. I love me some Pepsi from a fountain. It's not quite the same from a can. So I would be with maybe some of my friends who didn't answer are not tea people either. I am one with you, not really a tea person. So that's perfect. I know, yeah, it's the Midwest. Okay, so let's think about this. We got sweet tea, which most of us love, and unsweetened tea, which one of us at least would drink. When we are talking about sweet tea and unsweetened tea, what do we have more of or less of in these two types of tea? What's different between the two of them? Exactly. It all comes back to the sugar, right? So sweet tea is really sweet because it has a lot of sugar. Unsweetened tea doesn't have very much sugar, or a couple of my friends in previous semesters have corrected me. They've been like, the best unsweetened tea has no sugar, which is like, you are intense. Props to you. So sweet tea has much more sugar than unsweetened tea has in it. When we're talking about our types of tea, when we talk about the sugar that we put in tea, we would call that a solute. So this is a chemistry word for you, solute. When we talk about a solute, we mean something that is dissolved, something that is dissolved, a solute. So when we're talking about sweet tea and unsweetened tea, our solute is sugar. When we're talking later, when you're reading an article about plasma, the solute is going to be salt. How much sodium and chloride do you have in your plasma? Solute is just, again, a fancy chemistry word for whatever is being dissolved. Let's use what we just talked about related to these tonic words to see if we can fill in our blanks here. So when we talked about a hypertonic solution, what was the way that we described what hypertonic meant? Again, what was the way we talked about hypertonic? I bolded it, but I'm going to underline it. Yeah, hypertonic. Hyper meant more, and tonic meant concentration. Hypertonic, I have more of something. In particular, what I have more of is my solute. So when my concentration goes up, that makes my level of solutes go up. When I'm talking about a hypertonic solution, that's a solution that has more solutes in it. It's got more stuff dissolved in it. So when I'm talking about a hypotonic solution, what did this hypo part mean? What was going on with my concentration in a hypotonic solution? Yeah, hypotonic solution meant less. So in a hypotonic solution, that's going to be one that has less in it, or a lower number in it. Hypotonic solution. That leaves us with our last one that we talked about, isotonic. When we talk about isotonic solutions, they have the same number of solutes. It matches. Iso meant the same. So, two ways for you to kind of define these words. First way to describe what hypertonic means, we could say it's more concentrated. That's 100% correct. The other way we could describe what hypertonic means, though, is we could say that it has more solutes. It has more things dissolved in it. That means exactly the same thing as saying that it has a higher concentration. Those two mean the same. In a hypotonic solution, we can say that it has a lower concentration, or we can say that it has fewer or less solutes in it. Both of those things mean the same thing. And isotonic solutions have the same number of dissolved things, or they have the same concentration. Let's apply these words to the picture that you have in your notes. Can you help me out in the chat? What page do you all have this picture on? Okay, I'm seeing page 8 possibly. So perfect. We're on page 8 in the lesson number 2 packet. So when we are looking here at page 8, we're looking at something, literally its name is a YouTube. So if we were in person, I could crack the joke that you'd all groan at, like, this is the original YouTube. Isn't it awesome? And you would all groan and roll your eyes at me and not laugh. But I got to try to do some dad jokes, right? So we're looking at the coolest YouTube ever, obviously. Although it would be pretty cool if we were doing a science experiment with sweet tea and unsweetened tea. So it may not look really cool and in my little sketch here, but let's pretend that we're doing an experiment in my YouTube where I have the two kinds of tea that you could be drinking. You could be drinking sweet tea, is what most of us are drinking, or you could be drinking unsweetened tea. We just said on the previous slide that what is different between the two types of tea is the amount of sugar that we have in them. Not a trick question, I promise. Is it sweet tea or unsweetened tea that has more sugar? Which one has more sugar? Not a trick question, I promise. Yeah, it should be the sweet tea, right? Unless the catering staff got it mixed up at the event and they're passing out the unsweetened tea and saying it's sweet, right? I've been told that's the worst. I wouldn't know. I don't drink tea. So let's assume we got it right. Our sweet tea has more sugar than our unsweetened tea. Okay, so we got sweet tea over here. We've got unsweetened tea over here. We need to go to town then on my sweet tea side of this tube and add some sugar. Sugar is my solute. So I'm gonna get my little pencil here and we're gonna add some sugar to our sweet tea because that sugar is what makes it sweet. Hey, what do we think? Is that enough sugar or do we need some more? How much sugar do we like in our sweet tea? Yeah, lots of us are saying that looks gross. We need some more. Okay, let's get some more. Okay, we'll call this good. One semester I did this with a group of students and they weren't satisfied until I literally filled the entire thing with sugar. And I was like, oh man, who is gonna drink tea like that? Like I said, like the TV commercial guy, that is the recipe for diabetes, right? The diabetes guy. So we got some diabetes tea over here, and we've got our unsweetened tea. I know, it was so sad that he passed away. Yeah, we got Cane's tea. There we go. Raising Cane's tea over here on this side. Our unsweetened tea over here on the other side. The unsweetened tea will be hardcore, and we'll just say there is no sugar because we're like legit. Okay, so let's fill in our blanks here. We are talking about sweet tea. When I look at sweet tea, I see a lot of solutes. I see a lot of sugar compared to the amount of sugar that's over here in my unsweetened tea. If I have a lot or I have more in this solution, what tonic word means that I have a higher concentration on this side? What tonic word would I use over here? Yeah, absolutely. Several of us are chiming in. I would say that sweet tea is hypertonic. Hypertonic. Hypertonic means there's a higher concentration. Sweet tea is hypertonic to unsweetened tea because I have more solutes, more sugar in sweet tea than I do in unsweetened tea. So sweet tea is hypertonic to unsweetened tea. But check this out. I can compare the opposite way as well. Unsweetened tea that I see here on the right side has fewer solutes or no solutes compared to sweet tea. What was the tonic word that I used to mean that it had fewer solutes or it had a lower concentration? Yeah, absolutely. The opposite is hypotonic. Unsweetened tea is hypotonic to sweet tea. Okay, I want to pause for a moment and I want us to look at this. This is an important idea with these tonic words, with these concentration words. You really need to pay attention to the order that my solutions are in. Notice how when I start by talking about the unsweetened tea, and I'm comparing this tea, the unsweetened tea, to the sweetened tea, the word that I use to compare this one to this one is hypotonic. If I flip the order and I start with the sweet tea, if I start on this side and compare it to this one, now the word I use to make that comparison is hypertonic. It's going to be really important when you're working on the homework assignments, when you're working on the exam. Make sure to slow down and look at the order that your solutions are in. Because when you're doing the homework assignment, you're going to see numbers. It's not usually going to be sweet tea versus unsweetened tea, but you'll see some numbers. If I put a 5 and I put a 1, when I'm comparing something that has, let me get my pencil out here, something with a 5% concentration compared to a 1% concentration, the way I'd compare those things is I'd say it's hypertonic. But if I start with the 1% solution and I compare that to the 5% solution, the way that I would compare those is I'd say it's hypotonic. It's all about the order, just like it was with those directional terms in lab. Remember how much we loved or hated, more likely, those directional terms? It matters the order that we go in. So check that order to make sure that we're putting them in the right way. When we first started talking about tonic words, I put up a slide that said that diffusion and osmosis wanted things to be isotonic. Before we talk more about diffusion and osmosis, remind me what the word isotonic meant. What did isotonic mean? Isotonic. Yeah, isotonic means the same. Means the same. Okay, so let's chat about diffusion and osmosis. Diffusion and osmosis are two ways that I can make solutions that start out different, make them the same. So it's ways that I can take something that's hypertonic to its neighbor and turn it isotonic with its neighbor. I can make the concentrations the same. There are two ways that you can make the concentration of things that are different, make it the same. The first way that you can make different concentrations the same is by using the process called diffusion. When we talk about diffusion, we're going to move our solutes from one solution to the other. So when I'm looking at my tea here, I've got my sweet tea on this side. I've got my unsweetened tea on this side. When I'm looking at these two, it's obvious that they don't have the same amount of sugar. One way I could make them have the same amounts of sugar is I could take some of these little sugar molecules that are here in the hypertonic solution. I could move those sugars away. So in the process of diffusion, my solutes, or in this example that we're doing here, my sugar can move from one side of the tube to the other side of the tube. Diffusion would move that sugar from the hypertonic solution into the hypotonic solution to end up with isotonic solutions, so that the two sides of my tube have the same concentration of sugar. In the process of diffusion, I get these two halves of my tube to look the same by moving the solutes, by moving the sugar. So we already talked about this because we drew our arrows. When we're talking about using the process of diffusion to make sweet tea and unsweetened tea equal to each other, when we're doing diffusion, which way does the sugar move? Does it go into my sweet tea, or does it go out of my sweet tea if we want to make it equal, if we're equalizing things? Yeah, exactly. Several of us are chiming in. When we talk about diffusion, diffusion is when I move my sugar, and my sugar is going to leave the sweet tea, because the sweet tea has a bunch of it, and the unsweetened tea doesn't have hardly any at all. I'm going to share that sugar with the tea using the process of diffusion to make us have an equal number of solutes on both sides. So if sugar is moving out of the sweet tea to get isotonic, that means it's going to be moving into the unsweetened tea. We're moving the sugar into the solution that doesn't have as much. So now my unsweetened tea becomes halfway sweetened, and my sweet tea becomes halfway sweetened, and nobody wants to drink this tea, right? But at least it's isotonic. At least they're the same concentration. Too low or too high, depending on your taste. There is another way that we can make our concentrations equal. Instead of moving the solutes, instead of moving the dissolved things, we can actually move what's called the solvent, or the solution that the things are dissolved in. So here's my technical chemistry term, the solvent, what I'm using to dissolve things. In the body, we're going to assume that the solvent is water. In your body, that's the fluid that's going to move. So the other way to take two solutions that have different concentrations and make them the same is to move the water that's in one side of the tube into the other. Now, with osmosis, the movement of water is going to go to wherever there's a whole bunch of solute. In other words, the water that I have on this side of the tube, it's bouncing around, and it keeps bumping into other water molecules. And it says, man, y'all are really boring. I would really like to meet some sugar. There's not nearly enough sugar over here. I'm going to go to the other solution where there's a ton of sugar, and I'm going to bounce around and I'm going to meet all these new sugar molecules. So in osmosis, there's a ton of water over here where the water molecules keep bumping into each other. They actually go over into the other solution where there's less water comparatively because there's more sugar. And they bump into those sugar molecules and they feel much happier. Until we get to the point where water bumps into just as much sugar on this side as this side, we're going to keep doing osmosis. So in osmosis, my solvent, or my water, goes to where there's not as much water. Something that has a really low concentration of sugar, for example, so something like unsweetened tea, where there's very little sugar, that means that it feels like there's a ton more water. The water says, forget this. I'm going to go to where there's less water So in osmosis, the water moves away from that unsweetened tea and into that sweetened tea, because that side looks like it has less water. So let's fill in our chart here. We said with unsweetened tea that the water goes out of the unsweetened tea and it's going into the sweet tea. The goal being to get as much water over here to where water bumps into sugar and water bumps into water at the same rate over here compared to over here. This is a really important idea, and I need to pause for a moment because I need you all to stop and think this through. I need to know what questions you have, what you're thinking about these things. Help me out here. What can we put to words? Yeah, go for it, Fanchen.
Okay, my question is this is where I know I'm overthinking it, so I'm getting confused. With water, do you ever consider the water hypertonic to the solution when you're thinking about it? Or is it just, that's where I get myself confused, because I start thinking, okay, well, I understand there's more salt over here, but there's more water over here. So do you understand what I'm asking?
Yes, I 100% understand what you're asking because every semester students ask this. So thank you for asking this. This is a perfect question. So let me rephrase it to make sure I understand it correctly. So what Fanchen is pointing out for us is on this side of the tube, there is a ton of sugar. On this side of the tube, there is a ton of water. So when we're talking about concentrations, when we're talking hypertonic and hypotonic, how does that factor into the number of water molecules that we have? So here's the tricky thing with these tonic words. When we talk about these tonic words, we are always referring the number that you get for a solution. It's always all about the solutes. It's always all about the sugar. Or when we're talking about the cells in our body, it's always all about the salt. We kind of conveniently ignore the amount of water that's in these sides of the tube, except to say when we talk about diffusion and osmosis, here's a good way to think about both of these processes. In these processes, whatever it is that's moving, so whether it's the solutes, whether it's the sugar that's moving, or whether it's the solvent, whether it's the water that's moving, whatever moves always goes from a place where there's a whole bunch of it to a place where there's not a whole bunch of it. So this is why when we talk about osmosis, water will go from where there's a ton of water on this side to where there's not as much water. When we talk about diffusion, where the dissolved things are moving, the dissolved things are always going to go from where there's a lot of them to where there's a little of them. In these two processes, osmosis or diffusion, the thing that moves always goes from where there's a lot to where there's a little. But when we're talking about concentrations, when we're using these tonic words, or when we're using percentages, those things only describe what's going on with the solute. I always feel like I do a bad job describing it. Yeah, so follow-up question. Help me out.
Okay, so not necessarily a follow-up question. I just want to kind of put it in my own words and make sure that I'm clarifying. So let's take it, because like I see a lot of questions about putting a cell into a hypertonic or a hypotonic environment on the worksheets. So when you put the cell in a hypertonic, it's saying it's focusing on the sodium chloride levels. Is that correct?
Correct. Yes.
So even when we're talking osmosis, we're saying that it is moving into that. It is actually, if we're talking about the solvent, it is actually going to be in a hypertonic, even if you're talking about the solute being in a hypertonic, correct? So that when the osmosis happens, it's moving into a hypertonic because we're focusing on these solutes, correct?
Yes.
Okay.
Yeah. So let's look at where, because you were talking about the cells. So here's the image that you have in your notes outline. You'll see it on the homework too. Help me out in the chat. What page do we start talking about cells related to this hypertonic, hypotonic stuff? What page is this one from? Is this like page 9 or 10? Okay, page 10. Okay. So on page 10 is where the rubber actually hits the road, where we stop joking around about sweet tea and we start talking about the cells in your body. Remember how at the beginning of our class today, we said that your cells are filled with a solution. What's the name of that solution inside your cells again? What kind of fluid is inside the cells? Yeah, exactly. The fluid inside your cells is cytosol. The fluid outside of your cells has many different names, but in the end of this lesson, and as you're reading about what's called water intoxication, the kind of fluid you're going to be focusing on is called plasma. That's the fluid that's inside the blood vessels. When we talk about red blood cells, we can change the concentration of salt that's in your plasma. And we have an idea of what that will do to your cells, not just because we've studied it in humans, but because we've also taken some red blood cells and in the lab, and we've put them inside different solutions. So we've put them inside stuff that have different amounts of salt, and then we watched what happened to them. So this is a red blood cell. This red blood cell, notice that the amount of water that's going into the cytosol, that's what my yellow arrow means here, the water that's going into the cytosol is the same as the water that's going out of the cytosol. In other words, when osmosis sends some water in, osmosis also sends some water out. The rate of water going into and out of a cell, when that's equal, it means that the two solutions, the cytosol and what's on the outside of the cell, is isotonic. In other words, when I match what's on the inside to what's on the outside, no more water is going to move. The net movement, if I add together, so if I've got plus for what goes in and minus for what leaves, I end up at 0. If you take a cell in your body and you put it into a solution that has the same amount of salt on the outside as your cells have on the inside, this is what would happen. Your cells are happy, they're healthy, they love it. It's possible, though, to take your red blood cell and put them in a solution that doesn't have the same amount of salt in it. Let's start with this situation over here. In this situation, notice that water is rushing into my cell. There's this idea with osmosis, and this statement is in your notes packet. So let's underline, highlight, star it. The statement is water follows salt. Water follows salt, meaning wherever there's more salt, water is going to go there. So notice how water is entering this cell over here. If I've got a bunch of water going into this cell, that means that there's more water on the outside compared to how much is on the inside. That means there's more salt inside my cell that makes the water want to come inside. If I'm talking about the kind of solution where water wants to rush into my cell, I would call that solution a hypotonic, I'll do it all in caps, hypotonic solution. Hypotonic either means, it means two things. It either means I have less salt, hypotonic, less salt, or another way you can think about it is I have more water. A hypotonic solution has less salt, but it has more water. So in a hypotonic solution, if there's a bunch of water out here, that water wants to go into my cells, because inside my cell, my cytosol has more salt. My solution on the outside is hypotonic to my solution on the inside. I have more water outside, which means that my water is going to go in to my cell. The opposite is happening over here. Over here, notice that my cell is losing its water. So my environment must have more salt in it because water always moves in the direction that there's more salt. So if there's more salt out here, my concentration out here is going to be higher. I've got more salt out there. That means that the solution that's on the outside of this cell must be hypertonic to the cytosol inside the cell. Because there's more salt on the outside of the cell, the water that's on the inside wants to leave. The water's rushing out. So anytime I take a red blood cell and I put it in a hypertonic solution, so the stuff here on the outside has more salt or it has more sugar than what's on the inside. Anytime I do that, water is going to leave those cells. Those cells are going to shrivel up. They're going to get all small because the water that's inside of them wants to go out. It wants to go dilute what's on the outside, or it's really attracted to that salt out there. Anytime I put a cell in a solution that is hypotonic, in a solution that has more water, the water on the outside says, wait a minute, I see a bunch of salt in there. I'm heading in. The problem with hypotonic solutions is your red blood cells get all puffy because they're full of water. And if they get puffy enough, they might actually explode, which I don't think we have to have any anatomy knowledge to know that if I blow up a red blood cell, it's probably not going to do its job. It's going to be dead. So putting your red blood cells in a hypotonic solution where there's extra water on the outside, that water is going to rush in and it's going to make those red blood cells expand and possibly even explode. When I have those red blood cells in an isotonic solution where there's the same amount of salt and water on the outside compared to the salt and the water on the inside, this is where my cells are happy and healthy. This is where the amount of water that comes in is equal to the amount of water that goes out. And because of that, my cells are able to stay the right shape and do the right job. Big picture, Isotonic, good for your cells. Hypotonic plasma or hypertonic plasma, bad news. Not good for your cell either way. Either your cell is giving away its water or it's getting flooded with water, and neither of those are good things. So I'm checking out my question here. Yeah, so the question is in the chat is reframing it to think about it in terms of considering that there's only so much space, and that space is either always going to be water or it's always going to be the solute. Yeah, that's a great way to think about it. So either I've got a ton of water or I've got water and solute somewhere. And yeah, that's a great way to think about it. So no matter what, I'm always filling my space. Am I putting any stuff with that water, or is it just water? That's going to dictate where water wants to go. Water wants to be around somebody that's not itself. So if you've got a thing that's completely full of water, water's like, yeah, not so much. I'm peacing out. I'm going somewhere else. So yeah, great way to think about it, that there's only so much space, and either it's filled with water or it's filled with solute. I like that. Here is what I want to end our time with today. I don't think you have this in your notes. Is that correct? I'm pretty sure I made this up as a learning activity for us together. Do you have this in your notes outline? Oh, you do have it. Do you have it with glucose or do you have it with salt? I think you all have it with salt in your notes. Okay, so for my friends who already worked through this activity, this is slightly different from what's in your notes. So I'm going to challenge you all with this too, because this is new for us. In your notes, I told you the normal concentration of salt that's inside a cell. What is the normal percent concentration for salt, for sodium chloride? I'm going to make a note of that on our cell here. Normal cell salt concentration. Yeah, the normal cell salt concentration would be 0.9%. 0.9%. When you're filling out this activity in your notes, you are referencing, when we're comparing whether something is hypertonic or hypotonic to cytosol, when we're talking about salt or sodium chloride, your number inside the cell is 0.9%, 0.9%. So when you're receiving fluids, when we think about the plasma, there's something that on TV shows they like to call isosaline. Isosaline. For any of my friends who have time to watch medical TV shows, those were the days, says the mother. Isosaline is a saltwater solution that has the same concentration of salt and water as inside your cells. And here's our number for what the concentration inside is. So my hint for you as you're working on this activity in your notes packet, your number in the very middle of your cell is 0.9%. That's the normal concentration of salt. What we're going to do in class together to make sure we understand our tonic words is we're actually going to look at the concentration of sugar, of glucose. Now, I don't make you learn a lot of numbers. 0.9%, that is going to be a number to know. Let me circle that number for you. We do need to know this number. We do not need to know this number right here. This is the normal glucose concentration inside your cells. So inside your cells, we normally have a concentration of about 126 milligrams per deciliter. Not a number that I expect you to know. The only reason I gave you this number is because I want us to use this number to figure out all of these different types of solutions that we could put a red blood cell in, how do they compare to the solution that's on the inside? So let's say that I take a red blood cell with a normal concentration of 126 milligrams per deciliter of glucose, and I put this red blood cell in a glass of pure water. Pure water. My concentration of glucose in pure water is 0 milligrams per deciliter. When I'm comparing pure water to cytosol, which tonic word would I say that pure water is? Water is blank to cytosol. Which tonic word would I use to describe water? Yeah, exactly. Water is absolutely hypotonic to cytosol. Water is hypotonic to everything, to be blunt. The most water I can have in something is water. So a pure water solution is always going to be hypotonic to cytosol. If I were to put this red blood cell inside a hypotonic solution, inside water, which way does water move? Into or out of my cell? Which way is water going to go if I put a cell in it? Yeah, absolutely. I have no sugar outside my cell. I have 126 milligrams per deciliter inside. I'm going to have a bunch of water that's going to go into this cell. This is the situation that we looked at on the previous slide, where the cell swells up and it explodes. If we put a red blood cell in pure water, just water, no sugar in it, all the water is going to rush inside because they want to hang out with that sugar. This cell is going to expand and it's going to explode. Bad news. Okay, let's go down here. Dr. Aulis tried to do a little creative math here using the number of grams of sugar that's in a can of soda, a can of Coke, to convert it to our units here. So this may not quite be accurate, but the idea stands that if we were to take a red blood cell and put it in flat Coca-Cola, we let all the fizz come out, theoretically the glucose concentration in that Coca-Cola is 962 milligrams per deciliter. 962. And I'm slightly on the inside questioning my life choices, but not enough to change it. 962 milligrams per deciliter compared to 126 milligrams per deciliter. What tonic word would I use to compare Coke to cytosol? What tonic word goes in the blank right here? Yeah, absolutely. Completely and totally hypertonic, right? Which is fitting because like you're supposed to get a sugar buzz, right? So Coca-Cola is absolutely hypertonic to cytosol. Way more sugar than what I see in cytosol. Since this is a hypertonic solution, there's way more sugar outside than there is inside. Is water going to move into the cell or out of the cell? Which way is water going to want to go? Yeah, so water is going to want to leave the cell because the water says, wait a minute, there is way more sugar outside. I want to make friends with that sugar. I'm leaving. Peace out. And this cell is going to shrivel up. So you put a red blood cell in Coke, unsurprisingly, all its water is going to come out. It's going to go into that Coke. Let's go over here. There's a drink called Pedialyte. Pedialyte is what you give to a kid when, usually when they've been throwing up a lot. So it has electrolytes in it. It also has some glucose in it. In fact, its glucose concentration is 250 milligrams per deciliter. 250 milligrams per deciliter in Pedialyte. Compared to cytosol, what would be the tonic word we use to compare Pedialyte to cytosol? What word goes in this blank here? Pedialyte is, yeah, Pedialyte is hypertonic. It is still more concentrated than what's inside. Since it's still more concentrated than what's on the inside, what's going to happen with my water? Which way is that water going? If I put a red blood cell in Pedialyte, is it going in or out? Yeah, we're still hypertonic on the outside, so we're still going to lose some water. We're not going to lose quite as much water as we did with the Coke because it's not quite as much more concentrated, but we are still going to lose some water, absolutely. That brings us to our last one up here, and our last one up here is a 5% glucose solution. A 5% glucose solution has 126 milligrams per deciliter. It has the same concentration as the fluid inside your cells. What's that word for the same concentration? The same concentration? Exactly. Yep. So a 5% glucose solution would be considered isotonic. It's the same concentration inside compared to outside. And remember, when we put a red blood cell in an isotonic solution, a little bit of water thinks, hey, I want to go check out the sugar that's inside there. But at the same time, some of the water that's inside the cell says, hey, I want to go check out the sugar that's outside there. The amount of water that goes in is equal to the amount of water that goes out. So in an isotonic solution, your red blood cell stays the correct size. It looks normal. It can do its job normally. We just used our tonic words, hypertonic, isotonic, hypotonic, to predict what would happen to a red blood cell based on the concentrations of sugar that we could have around that red blood cell. When you read about something called water intoxication, that's our learning activity, the article you're going to read is called Strange But True, Drinking Too Much Water Can Kill. It's about what happens when we change the concentration of your plasma, when we change the level of salt that's outside your red blood cells. It's going to help you explore what happens in your body if we mess up this kind of chemistry. Keep in mind, regardless of what my numbers are, we could totally have changed this number. This number could have been 2,000 milligrams per deciliter, or this one could have been 30 milligrams per deciliter, or this one could have been 300. All you care about when you're figuring out if it's hypertonic, hypotonic, or isotonic, you're literally just looking at the numbers. We don't care exactly what the numbers say. We just care, is it higher or is it lower? Or is it the same? So when you're working on this activity with salt, the same kind of rules apply. The amount of water that moves, the direction it moves, that's exactly the same. We're doing it with salt. We're not doing it with sugar, but it's the exact same idea. I am promising you that on the exam, I'm going to give you something similar to this. So you know how your notes had salt in it and you practice this with salt? Today in class, we did this with glucose. On the exam, I'm promising you I'm going to give you something completely different. I'm going to give you a completely different set of numbers. You don't need to know anything about what ion I'm talking about or what compound I'm talking about. All you care about is how do these numbers compare. So this is what Dr. Ollis likes to call application questions. You don't have to know how much sugar is in Coke for sure, but if I know that this number is bigger than this number, I'm good. I can figure out which way water is going to move. Or if I know that this number is smaller than this one, I can figure out the way that water is going to move. So my goal with this activity is for you to practice thinking about what these words mean. Use the homework assignment to keep reviewing that. Use that article about water intoxication to be thinking about why this stuff matters. Remember that your lesson 2 homework for lecture is due by Sunday. Remember that we have a visible body assignment due tomorrow, actually a couple of assignments due tomorrow. And remember that we also have the lab assignment due Sunday, or the lecture assignment, excuse me, lecture and lab. I think my coffee didn't kick in. I think I'm about done. I need to stop talking. What questions do we have before we call it a day? Any last minute questions for me? Yeah, Kaylin's mentioning that she had water intoxication at one point. Yeah, too much water, too little salt. So get some salt to get balanced. Absolutely. All right, well, I am going to shut off the recording. I will stick around for a couple minutes if you have any last minute questions. Otherwise, remember that tomorrow I have open student hours for you to stop by. So I'm not going to plan a lesson tomorrow, but I'm available for any questions you have from 1 to 2:30. So feel free to stop by or feel free to send me an email. I will do my best to get in touch with you. Otherwise, have a great weekend. Good luck with your work, and I hope to see most of you or all of you back with us on Wednesday of next week.